Michael, for inviting me to return to my adopted homeland, second homeland, here in Galway, and really appreciate the opportunity to come today and speak on a lot of things that have changed within drug development and cancer. My world is both solid tumors as well as hematologic malignancies. I'm a pharmacist by training, but a clinical pharmacologist by conscription. So I work in various areas to try to understand drug disposition and patient variables that lead to that. There's been a pretty impressive change and sea change in the way that drugs are approved in the U.S. One of my areas of interest is immunotherapy, but also trial design and thinking about how we do that. If you look specifically at phase 1 trial designs and their classic purpose, the classic purpose was to determine safety, and still somewhat is to get the dose right, and you get the dose right by figuring out what the adverse events are as you escalate dose. And in general, the population in a phase 1 trial and a first-in-human study is a patient population that's generally got advanced disease, has pretty good performance status outside of their cancer being progressive, and really have exhausted all prior lines of treatment. And so they come into our unit, our program, and we offer them studies of drugs with letters and numbers, and then we figure out how they do. And we figure out how they do in a relatively short period of time, and oftentimes, at least historically, in a small number of people. So 15, 30 patients can dictate what the dose is going to be to carry forward, and that's going to be a theme throughout the presentation today, and certainly the phase 2 dose is the right one. So it answers the question, what's the right way to give a drug, and what should the dose be? Now, dose is the number one priority, and in my opinion, we don't do enough thought about schedule or sequencing. We certainly consider that in later stages, but in the phase 1 trial design world, we tend to not do that very much. And unfortunately, I think that's a pretty big drawback to a lot of what we do. And again, we also do many things now that we didn't do 10, 15 years ago, like multiple biopsies to understand pharmacodynamics of the drug. Did it get to the target? Did it inhibit a specific protein of interest, et cetera? And so we ask a lot of patients and other groups to try to help us understand the biology of the drug and the disposition of it. Classically, it's a 3+3 design with fixed cohorts, and so we have dose level 1, 2, et cetera, up to some number. The starting dose is based on toxicology, and so again, the adverse event world really drives what we do, and this is toxicology of generally 2 animal species. The animal species that's the most sensitive is the one that we use to select the starting dose. There's been a lot of discussion, and with an ethics colleague of mine and I, we've published on some of the ideas that we really start too low in many instances, and certainly those of you who work with monoclonal antibodies can appreciate that we start at minuscule doses and escalate from there, and you see 100-fold differences in doses from the first dose cohort up until the actual eventual dose. And many times we never reach the actual MTD in some instances. And then we schedule, again, based on animal PK and some toxicology, but oftentimes we base it on, well, there's neutropenia at day 8, therefore we can't give the drug more often than weekly or every 2 weeks. And that's, I think, a bit limited. So how do we get the right dose? We look at severe side effects in the first cycle, and that's also a bit of a limitation, particularly as we talk about drugs that come onto the market that are small molecule inhibitors, that are oral, that free up patients to do different things. Grade 1 diarrhea that lasts for 12 months is much worse than one single platelet value of 18,000 in the first cycle. So dose deliverability and dose tolerability extends much beyond the first cycle of treatment. And we think about phase 1 trials as being different in different populations. A dose-limiting toxicity, which generally is hematologic toxicity or some severe grade 3 or 4 non-hematologic toxicity, tends to drive our decision-making within dosing. But it can be different in different populations. If you have a group that's at risk of cancer and you're doing a chemoprevention study, phase 1 trials are performed in that population. So you need to make sure you don't do anything wrong, that your adverse event profile has to be incredibly clean for someone who only may get cancer, not who doesn't have cancer. When you look at a bone marrow transplant phase 1, it's a very different story. Engraftment doesn't occur by day 42, maybe certainly in AML trials, that may be a dose-limiting toxicity. That would never, of course, be allowed in someone with pancreas cancer on a phase 1 solid tumor study. And so the adverse event profile can be different. There are some major drawbacks to the way that we do these trials, and many times they're not, at least the first population is not the population of interest. If you have a phase 1 1 trial in a solid tumor population, it will be patients with pancreas cancer, melanoma, colorectal cancer, et cetera. And if you're going to carry that drug forward into a specific disease, you may not have a lot of experience within that. Similarly, patients may have multiple prior lines of treatment. And if your goal is to get to the frontline therapy option, then that's a different population in general. But we're deriving dose in these patients, and that's an important point to continue. What are the challenges of the historical paradigm was, as Jim just mentioned, it took a long time, and still does take a long time to get drug from first-in-human studies to patients in the clinic. And there are often disease-specific studies required after that initial phase 1 is done. There's certainly an increased cost of development. As you add on more and more trials and go in different directions, you add cost to the development of the drug. But in phase 1 trials, classically, once they were open, they were pretty inflexible. You had to do what you were going to do, and you may add an expansion cohort, but oftentimes a trial really didn't have more than 30, 40 patients within it. And it really it's really not a great platform for a first-in-human trial of a molecularly targeted agent. If you have a STAT3 inhibitor and you want to profile patients genetically for their presence of a STAT3 mutation, then a phase 1 dose escalation gets a bit wonky. It's hard to do that in a way that's going to be clear, and the enrollment can be quite challenging as well. And historically, when I first joined Emory in 2009, one of the first meetings I sat in was an argument with our billing people saying that, well, we can't bill insurance for phase 1 trials. I said, well, why not? I said, because CMS, our governing body, which regulates payment for Medicare and in some instances Medicaid, would say that, well, the primary objective isn't activity, it's tolerability. And so we're not going to pay for something that's not therapeutic. They saw these phase 1 trials as the equivalent of, a college student going into a CRO and doing a blood pressure test and blood pressure study and getting money for that and going out. So they saw it very similar to phase 1 trials in normal healthy volunteers, which is clearly not. And so luckily that conversation didn't last long, because I think I wouldn't have stayed for much longer if it did. So now what are we doing? Well, there's the complete phase 1 trial, and it's a development paradigm that's really changed substantially in the last 3 to 10 years, depending on how you look at it. Personalized, molecularly driven trials are very much there. The NCI in the US has something called the MATCH trial, and it's a platform where if you have a genetic mutation, there are many drugs and companies have lined up to give patients drugs that are selected for that mutation on that. And that's what's called a bucket or a basket trial. That's the NCI equivalent of that. It's incredibly complex and incredibly challenging, and I'll talk more about that as well. Disease-specific trials, integration of companion diagnostics early is certainly happening more and more in our phase 1 studies to try to predict who will respond, and perhaps more importantly, who won't respond, so we can look at other therapies for that patient. And then there are a number of novel designs that are being integrated I won't talk too much about. We've done a lot of work at our center with something called escalation with overdose control, or EWOC, which makes my kids laugh every time I say it. But it's a way to try to use real-time data within the trial to inform what the right dose should be. And so it goes as a very movable and malleable dose level. We're not fixed into these singular moments. We often use optimal biologic dose, and we jokingly say that rituximab never actually reached its MTD, it reached its FTD, which is its financially tolerated dose. There were, I think, 4 grams of rituximab were given until finally people looked and said, oh, look, we've saturated CD20. And we did that about 3 grams ago. So the optimal biologic dose is becoming more in favor, and it's certainly for monoclonal antibodies an important point. I think about the daratumumab example and the great PD work that's actually been done with Dara in terms of occupancy of CD38, and it's in a pharmacodynamic method. And then finally, we've talked about increasing the starting dose, because candidly, it's really tough when you look back at first-in-human trials. The first cohort never has activity and almost never has toxicity when you look at first-in-human studies. And so we've got to think about pushing that dose a bit more overall. So pembrolizumab, to my experience, is really the first drug that looked at this paradigm of a seamless phase 1 study. And so it was approved in 2014 in September based on a proportion of patients in their first-in-human study called the Keynote 001 study. And that study enrolled, or at least had within it, 276 patients with melanoma, many of whom were ipilimumab naive and some were refractory. And this gentleman here was treated in our unit with pembrolizumab. So, President Carter was someone who benefited from this really reduced time to approval of pembrolizumab overall. He's a really great guy. He's got a big belt buckle that says JC on it, and we jokingly looked at him and said, "Is that Jesus Christ or Jimmy Carter?" And he chuckled at that, but he does still teach Sunday school in Plains, Georgia. This is actually a picture of him receiving an award at the Carter Center one day after he'd gotten an infusion of pembrolizumab in our unit. So we're certainly looking more and more at seamless drug development. So there's a great editorial in New England Journal recently from the FDA looking at this idea of taking a drug and really bringing it all the way to development, all the way to market from a first-in-human study, and really just altering that study and morphing it as you go along. And if you look back, there's been a lot of blurring of the phases of trials. We've had a lot of drugs approved in the U.S. on phase 2 data. Interestingly, bendamustine was approved on phase 2 data. A lot of people don't realize that. Crizotinib and osimertinib, both pretty major advances in the treatment of lung cancer, were both approved on phase 1 and 2 data. But if you look at pembrolizumab as the poster child of this, the phase 1 was started in 2011, and the early activity that was seen in melanoma and lung cancer really led to a massive expansion of cohorts. And so rather than doing a separate phase 2 or a single-arm phase 2 or a large phase 3, they just began to morph this study into a way that greater and greater numbers of patients with a specific disease were treated. And the total population for this phase 1 trial was 1,200 patients, which just makes everybody kind of laugh, because classically phase 1 studies are no more than 30 or 40 people within them. But this trial was certainly a large expansion group, and it led to approval in melanoma, non-small cell lung cancer, and a companion diagnostic of PD-L1 testing in lung cancer. And currently 40 trials have been identified by this FDA group that are pursuing this same approach. And when you look at it, it's a great idea, and a lot of good things can come of it. But there are real questions around this design and the way that we can really understand how this drug is working in many, many ways. And so if you're a company, these are some of the questions that FDA had asked. You can read these yourselves, but there are a lot of questions around the ability to detect a difference, sample size concerns. If it's going to be used for regulatory approval, make sure that it's there. Are you updating the informed consent in a relatively frequent fashion? You're learning as you go along, so you can't just hold on to that knowledge. You have to share that knowledge with potential subjects coming into the trial. Is that happening in real time? Is that something that you're committed to do as a company as you begin to develop it? And this is the new phase 1 trial. The drug names have been blacked out to protect the innocent, but this is a trial that we are conducting at Emory, and you can see that in part 1 there's a number of combinations of drug plus drug 1 at a fixed dose of 240 with drug 2 at escalating doses. And over time, each of these cohorts is a separate cohort. And so you're escalating up to a likely, based on the adverse event profile preclinically of this compound, we're probably going to get to 800 milligrams. And so this is a minimum of 3 patients per cohort. But then we're just going to blow it up in part 2 and look at all of these diseases on the right as a way to try to understand its initial activity. And the likelihood is that if one of these comes out positive, and my prediction based on preclinical work is that it's going to be cervical, head and neck, and bladder as a big way, that you're going to just start to really expand those cohorts, and you'll have another example of the 1,000-patient phase 1 study. And then, this is my area of interest in clinical pharmacology, and so part of this is that there will be a sub-study within this trying to understand food effects within this space. This is a heme example of another compound, a first-in-human kinase inhibitor that will also remain nameless, but similarly here, this is multiple treatment groups based on disease, and then there will be expansions beyond that, and this isn't even the entire study. This is just the first sort of third of it. So these trials are coming in more and more, and it does create a bit of a challenge. And so if you look at seamless drug development, there are still gaps in what we're looking at within the data that's presented. The complete phase 1 asks questions like, when do you do clin pharm studies? And I'll talk a lot about that in a second, because to me, we're not doing the best job of clinical pharmacology understanding of many agents. Well, what about a control group? If you're trying to understand adverse events in a population, then you need to somewhat have a control group of a disease either treated with conventional treatment or untreated to try to say, all right, well, what's the adverse event of the drug compared to a natural biology or a different group, so that we can really understand treatment emergent adverse events. Again, the personalized, disease-specific, and molecularly driven trials within our center has become a bit of a challenge. Who should own these trials? Where should they go? And then there's a lot of effort for rare mutations. We have a compound in-house now where the hit rate for the genetic mutation was unknown as we came into the study. And as we've gone along in the study, it's pretty clear that the hit rate's about 1 in 20. That's a lot of patients that don't have the hit rate if you're looking at a broad population of solid tumor patients. And so we're left with a lot of effort of research staff to get tissue, send it off for testing, and more importantly, patients are waiting for those results and may be negative for them. And so the molecularly driven world for me is a bit unsettling in many ways, and I think it's continuing to become more unsettling rather than less. So this is our traditional approach to dose finding, and so phase 1 and 2 really sets the foundation for what the dose will be for all trials carried forward, all evaluations carried forward, and you determine that dose for registration studies. And then once you get into the area that I believe is the best time to really understand clin pharm, when you're giving the same dose to a number of different patients. We don't really do that in the development world. We tend to not really capture a lot of endpoints like pharmacokinetics, pharmacodynamics, et cetera, because the signal and the cost is high, but the signal is something that we're trying to get done pretty quickly. And then commercial access. And so a number of things that have happened with FDA's accelerated approval of drugs is that we're doing trials that we would have done before approval, after approval. And that matters because if you're a patient walking in the door and you're being offered a new kinase inhibitor for third-line ALK-rearranged lung cancer, and you can't get this drug unless you go on to the trial, and it's a food effect study, you're likely to go on to the trial. Take that moment post-approval. You can get this drug from your treating oncologist on prescription. Your insurance pays for it. You have no really excitement to go on to a food effect study. And that's a problem because we need to understand drug interactions, transporter interactions, food effects in hepatic dysfunction, renal dysfunction as soon as we can, because there are patients that will need that, and certainly the food effect is one of the biggest issues. And so if we don't know that early on, then it's very hard to find it out afterwards. And we're going back now, along with a trainee of mine and I, and we're looking at post-approval commitments of drugs since 2000 to see how many of them have gone unpublished and undone. And it's a lot. It's more than you would potentially think. And this slide just depicts that accelerated approval world. And if you look at the years 2006 to 2010 in the US, those are the oral agents that are approved. I'm going to focus on oral therapies for a bit now for the rest of the presentation. And really, things were ramped up impressively in 13 and 14, and certainly last year was an incredible year. Gefitinib was already out there, but it was reapproved in the US based on EGFR mutations. And so all of these agents on both of these columns were FDA approved, and they're all oral. And then finally, so far we've got venetoclax for approval this year. If you think about an oral medicine and giving it, there are lots of things that come into play that aren't in play with a monoclonal antibody or an injectable otherwise. There's a lot of variability. One area that's become another passion of mine is the inability of companies to do a great job of pharmaceutics. Pretty basic stuff like pill size, pill burden, bioavailability, things that allow us to understand in a better fashion how drugs get absorbed. The effect of concurrent agents and foods. There's certainly a decreased interaction with the healthcare team when you give an oral therapy, and that's good. If we turn cancer more into a chronic disease, patients will continue to live. President Carter will continue to go and get awards and do Habitat for Humanity work, et cetera, and we want that. But it does create a bit of a challenge in terms of understanding how patients are doing if you don't see them as often. Copays and access are still an issue. Adherence may be an issue in some instances, not just patients not taking the drug, but occasionally patients taking it incorrectly or too much of the drug. We've had patients take lenalidomide continuously. We've had people not take breaks, and so that's where schedule becomes important, because if you continue to develop pretty hard regimens for patients to follow, you're going to increase the likelihood in a patient population that has other comorbidities that they're not going to take it correctly. There's population PK variability. Absorption is certainly an issue. We have germline distinctions between us based on how we absorb drugs, how we transport drugs into the liver, how we metabolize drugs. The cytochrome P450 system, phase 2 metabolism with glucuronidation, et cetera, may all be germline variants and potentially different between different people, different sexes, different races, et cetera. Distribution can change over time. Certainly the obesity epidemic in the US and worldwide has become a bit of a challenge in terms of thinking about where drugs go, where they distribute within fat or not. If they do, there's a depot effect, et cetera. How do you dose based on total, ideal, or other weights? In some instances, metabolism can be variable. Again, genomic differences, hepatic dysfunction, concurrent agents. And then finally, biliary and other routes of excretion can be different based on the presence of a gallbladder or not, et cetera. And so we can have lots of variability in how patients handle drugs. And finally, the assay performance may be different as well. And so with oral drugs, you think about phase 1 development, and this is a classic paradigm. So this is a perfect pharmacokinetic concentration time curve. And so we've got a patient taking a drug at the same time every day, and look, this is a nice, beautiful sawtooth pattern that we all like to see in the clinical pharmacokinetics world. And so we ask questions in phase 1, but we really alter the dose. And so we keep the interval consistent and say, all right, well, what happens if you give 1/4 of that dose, and how does the exposure overlap? Because it's really, dose is kind of a funny thing. I mean, I'm a pharmacist, so we're often sort of patting ourselves on the back when we calculate a BSA and we give a number that we've multiplied by the BSA for the final dose. And we really get excited about carboplatin dosing because, we're using lots of math to do it. Well, the math is really not very helpful in most instances because the exposure is incredibly variable patient to patient. Well, let's look at a patient's experience with this graph. So patients will take the same drug, every same dose of the drug, but they won't take it at the same time every day. So this is a patient's experience of a clinical pharmacokinetic profile, and that's that, oh man, it's 12 o'clock. I still haven't taken my nilotinib. I'm really hungry. I'm going to go through the Burger King drive-thru and take it with a croissant sandwich. And oh look, my peak concentration is 4 times what it was if I didn't take it with a crust sandwich. And then oh look, my QT interval is now 550 milliseconds. That's all bad, by the way. And so patience and understanding those variables is incredibly important because that's what patients do. Patients don't look at this beautiful little graph. That's not what they do. They don't care. They take their drug when it fits into their lives, as they should. And so it's our job and responsibility as researchers to make that as safe a process as possible. And we want to do that because of this relationship. And I'll focus specifically on the idea of a therapeutic concentration in adverse events. And oftentimes this is what we see. We see a flattening of efficacy based on dose. So the more you give, for those transplanters in the room, this is heresy, but more is not always better. And so you certainly can get a flattening in most instances of effect. You can also get a flattening because of PK parameters like saturable absorption. If you give vismodegib, you can give 1,700 grams of vismodegib and you'll only absorb 200 milligrams of it. And so there are specific things as well. But certainly as you get more exposure, we do see a lot more toxicity. Food effects, high-fat meals, organ dysfunction, drug interactions with things known and unknown. St. John's wort's the bane of my existence, as well as other natural complementary therapies, because we don't really have great understandings of how they work. So we do this through preclinical toxicity data, formulation, phase 1 dose derivation, and oral drugs are a little bit harder to figure out because of formulation size. We can give 1.3 milligrams per meter squared of bortezomib, or 1.2 or 1.6, but we have to give sunitinib at 12.5, 25, et cetera. And so formulations come into play because they're fixed and we can't vary them as much as one might like. The MTD is often the only dose evaluated later on, and we define dose for a population, not for an individual. And that's an important point in the clinic, that we have a dose, and it's almost like a confidence interval, or 95% confidence interval. Perhaps this dose works for the middle third of people, but there will be people on both sides of that curve that we get it wrong. We can get it wrong when we know there's an adverse event, the exposure is too high, and we can dose reduce. My concern as a pharmacist and pharmacologist is the other group. That's the group that their concentration may not be high enough due to a variety of reasons, whether it's adherence or other things, and their cancer progresses. And we just sort of say in the solid tumor world particularly, well, their cancer progressed. And we never really asked that question, did they get the right dose? Did they get the right exposure? CML's done that fairly well compared to other diseases. And so the limitations are really there. Our dose-exposure relationships sometimes aren't greatly defined. And there are a number of drugs that are getting FDA approved that kind of drive me nuts. You look at the phase 3 data, and you'll see that in some instances up to 85% of people need a dose reduction in cycle 2 or 3. That means we're getting it wrong. Something's not happening correctly if you're having to reduce 8 out of 10 people on a dose. And so the optimal biologic dose may not be the MTD as well. Chronic therapy-limiting adverse events aren't great because, again, grade 1 diarrhea that lasts for a year, grade 2 nausea that lasts for a year is much worse for a patient than an early first cycle adverse event. And we rarely explore multiple schedules in a comparative fashion. I think that's something that we need to do a bit better as well. So what does it matter if we get it wrong? Well, if we focus on rapid development, which we should, I'm not saying we should not try to get drugs to the clinic faster, but if we get it wrong, we're going to have major problems in terms of use after approval. And so these smaller sample sizes, even if you look at breast cancer, for example, and a new drug in breast cancer, most of the time in trials historically, for example, trastuzumab, it would have been used in anywhere from 800 to 1,000 women. These days, trastuzumab would probably only be used in 200 to 300 women. And so even though that's not the full population that's going to get the drug post-approval, it's still a lot more than it will be with our current paradigm. And so we've got to be mindful that post-marketing adverse events and pharmacovigilance may become even more important than they are now. We may or may not think about comorbidities and performance status. The average age of a trial patient, regardless of disease, is about 9 years younger than the actual patient in the clinic. So we're not even really reflecting our patients in the clinic very well. I'm part of an ASCO group to try to look at eligibility criteria and modernizing them, because the other issue with the phase 1 complete trial is that if you start a trial and say, well, we really don't want anybody with a creatinine clearance less than 50 coming onto this study. In myeloma, that's horrific. You're going to exclude a ton of patients in the relapsed refractory setting. And you just can't do that. Now, you can do that initially, but then go back and retool eligibility criteria as you learn about pharmacokinetics, tolerability, and other things. Excluding patients based on preclinical data who have had an MI in the last year. In the U.S., that's like a third of people over 60 these days. So you can't do that. I mean, it's just not the real population that's going to be treated. I don't think I'll do that if that's all right. So this is a fun study. This is a case study of a drug. It's a multi-kinase inhibitor approved in 2012. This is audience response time. The FDA-approved dose was the maximum tolerated dose from the single-agent phase 1 study. In the phase 3 trial, this is that agent. 86% required at least one dose modification. So it was either an interruption of treatment, a dose reduction, or a complete stopping of the drug based on tolerability. And 2 dose reductions were allowed within the study, down to 70% of the starting dose and 40% of the starting dose. This is where I get excited and most people go to sleep. So the PK and PD of the drug, a high-fat meal, a stick of butter, increases the area under the curve by 57%. So that's a pretty big change in exposure, and that's what we look at as a measure of drug exposure within the body, not just dose. The half-life is 55 hours. It accumulates 5-fold at 21 days. So if you're taking a dose today, you'll be getting the equivalent of 5 times that dose in 3 weeks. If you look at a phase 1 trial and the cycle is 3 weeks long, you've already expanded beyond your dose-limiting toxicity period of evaluation. So you're already behind the 8 ball at 21 days. There's no relationship between exposure and progression-free survival, and that's shown here on this curve, that if the dose banding of the patients on this study anywhere from 20 to 120, this was the PFS. Anywhere from greater than 120, this is all the same line, by the way. I don't know if you noticed that, but there's no difference. So the exposure doesn't relate, the dose doesn't relate to progression-free survival, and similarly on a PK side, the exposure doesn't relate to progression-free survival. On an adverse event side, the increased exposure is the decreased time to first dose modification, so the more you have on board, the more likely you are to require a dose reduction, and you do have a QT prolongation of the interval with the drug. And there was a post-marketing requirement by FDA FDA to study lower doses. Does anyone know what this drug is? It's cabozantinib. So cabozantinib is approved in the U.S. originally for thyroid cancer at a dose of 140 milligrams. The manufacturer of cabozantinib and everybody else realized that that dose was way too much for anybody, really for thyroid patients. And so it was marketed as a tablet at 140 milligrams. They studied it in renal cell, and they studied it at 60 milligrams, and it beat the competitor in their comparator trial. And so they market it under a different brand name in a capsule. It's the same drug, but they just got the dose wrong early on. And this is some of the consequence of carrying drugs forward too early, because the phase 1 dose was 140. That came out of the dose escalation. And so we're getting it wrong. Think of carfilzomib. We're not dosing carfilzomib correctly if you use the label. We're not dosing a ton of drugs correctly if you look at the label. And we just looked actually at our experience in the southeast of carfilzomib, and the vast majority of clinicians actually, or physicians, don't escalate from 20 to 27. They don't even follow the product information. And so getting it wrong early is really a big challenge in many, many ways. And so this is the fallacy of cycle 1 toxicity reporting. Phase 2 doses come from grade 3 or higher toxicities. The limitations are certainly listed here. Capecitabine is a classic, and so capecitabine in breast cancer, nobody uses 1250 per meter squared twice a day anymore. That's way too toxic for the vast majority of women. But yet this was what was carried forward. And so a group out of the NCI looked and said, well, what about this cycle 1 issue? And how can we use this data to perhaps improve the way we think about dose? So they went back and looked at 77 trials and looked at time to toxicity and looked at when that toxicity occurred and its worst in any given patient. 40% did develop it in the first cycle, so that's good. 4 out of 10 patients have their worst adverse event in the first cycle of treatment, but that means 6 out of 10 didn't. And so if you think about the first cycle being the dose-limiting toxicity period, that means that we're not capturing the majority of those individuals. The duration is listed here as well. And they focused in on 3 organ sites of toxicity, the vast majority of which are GI, but there was some rash and renal dysfunction as well. So this was their data when they looked back, and you can see, I'll point out specifically that a definitive interruption of treatment occurred at cycle 2. And this is the mentality of the phase 1 person, and I am in this camp. You'll have somebody who's on a trial, they're in week 3 of 4, and they're having a side effect, they're having an adverse event, and we'll say, well, let's just get them through the first cycle. Let's just get them through, and hopefully it'll be okay, or they'll abrogate to it. And that's true of certain drugs like panabinostat, for example. Patients that can tolerate it after about 4 weeks, they're probably going to be okay, but that first 2 to 6 week period can be kind of rough in many, many instances. And so the problem, though, is that if you're using this first cycle for dose, you're not capturing all of these important events that are occurring later, but most importantly, those in cycle 2. And so this was their model for thinking about how a phase 2 dose might be derived from this information. If the early toxicity is equivalent to the later toxicity, you've got it. You've got the right dose and you can move forward. But if it's not, then you need to think very differently about how you consider next steps for dose. You can do one of 2 things, and in these days you would just add an expansion cohort that's much larger that would use that same dose, or perhaps 2 expansion cohorts in the best design, and one would use one dose and one would use another. Because now put yourself back in the patient's shoes. The FDA mandated that cabozantinib do post-approval studies. You're walking into your thyroid cancer treater, and they say, hey, I've got this study for you. It's of a drug, cabozantinib, and it was FDA approved at 140 milligrams, and we'll randomize you as part of this study to get either 140 or 60 milligrams. What do you think? The silence will be deafening. I mean, patients believe that more is better. And so you're just never going to accrue to that trial because you got the dose wrong. But it was an FDA mandate, and that's great, but it's never going to work. And so you can't actually find these things out. And that's, I think, one of the challenges of moving into this seamless paradigm is that the early stuff matters. It really matters. And if you don't get it right, it's going to be wrong for a long time. So I was speaking with Michael yesterday and the evening before, and I thought it would be helpful to share a little bit about our work at Emory and bringing forward a phase 1 program. And so I joined in 2009. And at the time, my mandate from my boss was create an infrastructure and administrative support for conducting phase 1 studies. And so when I came, I gave this slide as a talk, and I think it's an important slide because it still resonates in many, many ways. If you're going to have a successful clinical trials program, you have to have a clinical trials culture. That culture can be simplified as you have a trial for every patient and a patient for every trial. And if you can do that, if you can get to that place, then you're in a really, really good place, because you're able to then offer therapies and ask questions that are important to help those with the cancer that you're focusing on. And so that culture's got to be sort of inundated everywhere. It's got to be at the level of the regulatory support, the person collecting the lab sample, the nurse delivering the care, and the administrator looking at the budget. And if you don't have that culture all over the place, it's really a hard road to go down. So this is the opening of our phase 1 unit in 2010. You can see my former boss, Fadlo, is scared because I've got scissors, and we're cutting the unit to an 8-bed, or 8-chair, 4-bed unit at the time. Let's see, some of you may recognize this guy, much younger, much more attractive than. He's my current boss, Sagar Lonial, who's done a lot of work in myeloma over the years. And so it's really been a great collective group and effort at Emory to bring this forward from those days. And so we have a unit. The unit there is purposed for specifics of centralized care. Sponsors love it when they come in. I say, hey, look, hey, Jenny, hey, Carrie, how are you guys doing? They've been nurses in the unit for 5 years. They know this. They get it. They understand adverse events. They understand how to give drugs. They understand not to flush the line of the monoclonal antibody at the end of the infusion and make a peak concentration that's 4 times what it should be. So these kinds of things are ingrained in the staff that are there. All of our study-related procedures are in the unit. I live in the unit. My office is there, along with my work wife, another good person of Irish descent, Colleen Lewis. And so we are in the area all the time, seeing patients, talking to them, understanding subjective adverse events. The lab stuff's easy. A neutrophil count's easy. Frequency of diarrhea, severity of diarrhea, what did you do for it? How many times did you take that? Did it bother you overnight, etc.? Those kinds of nuances are certainly in our wheelhouse. And so we have lots of vital sign machines and EKGs, et cetera. Your facility here is beautiful. It's actually a great facility as well, and I'm somewhat envious in some respects. We have a lab right next to it. It's 200 square feet of -80 and hood and et cetera, and so that's been nice as well. We have good data capture. We have lots of people running around doing lots of things. Our unit is an incubator for many practices in nursing. For the general infusion center, it's an incubator for training of junior physicians on how to conduct trials, because if you come to our Tuesday morning phase 1 meeting, there are about 15 physicians around the table. They all represent different disease groups, and none of them will say I'm with the phase 1 program. They'll say I'm with GI, I'm with melanoma, I'm with sarcoma, et cetera, I'm with myeloma, but they're all part of the phase 1 program. So it's a very matrixed group in that way, and it's really a nice learning environment for people to figure out how to do trials. So I'm the director. We've got a lot of people there. It's a lot of folks that have grown over time. My research team is now 7 in terms of regulatory coordinator staff. We don't have nurses don't want to go into research in the U.S. It's been incredibly difficult to find a good research nurse, and so we've really run with our group of coordinators, and this is our space. And this was my mandate at the end of the day, because the NCI wants to see you get better in terms of your accrual to patients on trial, so you're answering important questions. When I came in 2009, this was the or in 2007, this was the trajectory. And so it sort of went up. Certainly in 2004, phase 1 work was incredibly unique and not done at Emory, and over time we've continued to escalate that. I only go out to 2011 because this number is consistently between 175 and 200 now, out to last year. And so we enroll we enrolled 183 patients on phase 1 studies last year, and that's probably where we are as a center. When you go to MD Anderson, that number is more like 1,100. When you go to Sarah Cannon in Nashville up the road, that number is about 600 to 700. But we would have to blow out a lot of walls and hire a lot more people if we were to reach those numbers anytime soon. So what did we learn as we did this? Well, we learned that we needed to do a better job of writing a grant for the UM1 to become part of the CTEP group. We're now with Hopkins, joining them in their UM1. everyone, molecularly defined trials, be very thoughtful if you consider doing these trials, because it is a lot of work. And it's a lot of work that may not yield therapy for a patient on a trial. It's important work, but I'm candidly becoming more and more disillusioned at molecularly driven and molecularly selected therapy. We need better pharmacoanalytical resources. Radiologic review is important. Using RECIST criteria is something that's evolved over time in our group. And the bottom point is probably the one that I had to learn the hard way. I went to every single disease group leader, and not everybody's going to be engaged in phase 1. And that's more on the solid tumor side than the heme side, to be honest. But candidly, we just don't get breast cancer patients referred to our program. And that's the nature of the treatment of the disease, but it certainly would be nice to do that. So with that in mind, I want to thank everyone. I want to thank Michael for inviting me here to come to Galway. I think sometimes this is what it feels like we do when it comes to drug development and drug design, and this is the cast of characters that I get to work with. So thank you.
So thank you very much for that really great elucidation of the challenges we have in front of us, but also some of the things that we can learn from the pharmacokinetics for patients that are already in the clinic as opposed to in trials. Has anybody got any questions? Prof O'Dwyer?
Well, a great talk, Donald. Thank you very much. I guess my question is specifically in relation to the example you gave with the multi-kinase inhibitor, which I'm not going to try and pronounce. But they obviously went for the maximum tolerated dose and brought that forward. Was there much in the way of pharmacodynamic assessment in that study, and did they actually find that they were hitting the target at a lower dose, but decided nevertheless to go forward with more is better?
That's a good question. It's a dirty kinase inhibitor. I love the idea of targeted therapy. Busulfan is more targeted than cabozantinib. I mean, these targeted therapies aren't targeted. Monoclonal antibodies are targeted. These kinase inhibitors are not targeted. So they looked at the wrong protein. They looked at RET, and they said, all right, phosphorylated RET. Oh, look, it's at 99% at 140, but it's only at 73% in lower doses. And so they felt like that RET was the driver for that. It's probably not. It might be in thyroid cancer, so I'll give them some credit in terms of thinking about 140 as the dose there. But it inhibits many other small molecules, or many other proteins. So they ran with their PD data and probably shouldn't have. So that's a hard lesson, and that's a hard thing to know in the prospective fashion.
Thank you. Dr. O'Connor?
Thanks, Donald. I really enjoyed your presentation, more because I'm a fellow pharmacologist myself.
Nice. Stay strong, brother.
Yeah. A couple of questions, if I might, actually. Briefly, your thoughts on modelling. I saw you mentioned statistics and that in there, but you didn't really talk about whether you use modelling and try and, I guess, be predictive out of your patient data.
Yeah, so PK modeling or Bayesian statistical modeling. We don't use a whole lot of PK modeling, honestly, and a lot of that is because we don't have real-time data as we look forward. Now, if you're referring to the clinic, I still believe in therapeutic drug monitoring. I still believe in pharmacokinetics and its power to make dose right for the population. I believe in pharmacometrics, et cetera. But I'll tell you that I tried to do an everolimus therapeutic drug monitoring trial, and it was a small study of 22 patients with labeled indications of breast cancer, renal cell cancer, and neuroendocrine tumors. And the time that I opened it, my dear friend and colleague Ruth O'Regan left to go to Wisconsin, so the breast cancer referrals dropped off. It was hard to get patients onto the study because it's, again, a post-approval kind of question. So I wanted to make that happen, but it just didn't come about. And I think we still have a role. I think actually monoclonal antibodies can be an interesting question for therapeutic drug monitoring as well. But from a modeling perspective, I haven't really approached that from a real-time basis outside of that trial.
You put an interesting teaser in your closing statement, probably by no accident, where you talked about frustration with the molecular therapies. Do you want to say a little bit more on that? And does economics come into that as well as the dirty effectiveness of some of these drugs, or what's your frustration there?
My frustration is probably fourfold, and I'll try to keep my comments brief. But the frustration on the one hand as a pharmacist is the pharmaceutics. Abiraterone is a drug that you have to take 4 large tablets every day, and it's 1,000 milligrams. If you're a melanoma patient with a BRAF mutation and you get placed on vimurafenib and dabrafenib, you're taking an extra 10 pills a day, twice a day. If you're taking erlotinib, you can't take a proton pump inhibitor. I mean, these are simple things. I mean, these shouldn't be that difficult from a pharmaceutics perspective to get right and to get better. So that's one side of the kinase inhibitor world. The other side is that we're trying to turn everything sometimes into an orphan disease. And so, let's say you're a TRK mutation patient, TRK2. There might be, in salivary gland cancer, it's about 80-90% of people. But it's not, they're present in many, many others. And so if you're a company marketing that and developing that, you've got a very small market to work with. And that's going to drive your price because you have to charge more to recoup or to get what the market will bear because the market is smaller. We could have personalized bortezomib based on 20S proteasome activity. I mean, it could have been, there are the ways you could actually personalize lenalidomide with cereblon. And I'm worried we're going down the wrong road personalization with PD-1 antagonists. I mean, it's a little bit like pharmacogenomics. What kills pharmacogenomics is a better drug. So you don't need to worry about VCOR mutation with warfarin in the US anymore. You just give them a NOAC or a DOAC or whatever you want to call them now. And so, better drugs kill personalization. And I think that's good. A good drug that works in everybody is better than all the heavy lifting of figuring out genetic mutations for the right drug for that patient, rather than saying, oh, look, let's just give them pembrolizumab because it really works and we don't have to think about it. And that's better. I mean, it really is better. This personalized therapy is driving me nuts on the solid side because mutations, we're not curing anybody either with small molecule inhibitors of BRAF or ALK or EGFR. We're giving patients better quality of life, and that's important. But resistance always comes. And so putting all of this effort in personalized therapy is challenging to me.
So one of the big challenges is actually to find the first starting dose in a phase 1 trial, and then moving the drug from preclinical, where you know what is the active exposure or the toxic exposure into humans, is a big challenge. But what do you think about microdosing experiments before actually doing the first trial in order to decide starting point?
I think it was a good idea when it came out, and it's been really not carried to fruition. The first phase 0 study that I saw published was ABT-888. It was done at the NCI. ABT-888, talking to them. So this concept of microdosing is giving incredibly low doses of a drug as a single dose to people with cancer so that you can optimize your assays, whether it's pharmacoanalytical assays or pharmacodynamic assays, and really just try to tune that up before you go into a phase 1 trial. So at the NCI, where people come to get care for free, in the NIH, they had to offer that phase 0 trial to 10 individuals before one would accept. There's no possibility of therapeutic benefit. There's really a low likelihood of adverse events. And so it's a bit more like a normal healthy volunteer study than it is a phase 1 trial. You're not going to get a lot of buy-in for that. You can see that in the number of trials that just haven't come to fruition that are phase 0 studies. There are good populations to do that. Perhaps the adjuvant population that's waiting or is post-resection, post-chemotherapy for their disease. But it's really been a challenge to get conducted.
Could I ask one question? It's actually in a way a little bit related to that, but it's the concept of what you think of intra-patient dose escalation, and whether that's something that can or shouldn't be used.
I love that idea, and we did it. So ixazomib, we performed the phase 1 study at Emory. A gentleman who I'll always remember was on the second cohort of that study. He'd had stable disease for 8 months, and we'd cleared cohort 4. And so we said, look, this guy needs to go up. And intrapatient dose escalation was clearly prohibited within the study. So I emailed the IRB. I emailed Takeda and said, look, you guys have data. You've cleared cohort 3. It's an oral proteasome inhibitor. It's not, we're not going to see some late cardiac effects and then have to figure out, was it this dose level or that dose level that caused it? And so I lobbied hard, and we ended up being successful in that individual. And we bumped him up, and he had a little bit more nausea, and he went into a PR, and he stayed on for the next year and a half. So, that was a great example of a time when it was a good thing to do. The challenge is just what I mentioned, that chronic toxicity. That if you have a patient on cohort 2, you escalate them to cohort 3, and they develop neuropathy, or they develop a more chronic adverse event, you've lost the ability to dial in, was it this dose or was it that dose? And oftentimes we just don't know if more is better or not.
Okay. So on that note, I'd like to thank you very much indeed, Dr. Harvey.